Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Naufal, and today I'll be covering about one of something that is crucial in our body. So crucial, you might can't imagine life without it. Can you guess what it is? Of course you can. It's already stated. It's already stated here. Yes, it is enzyme. So what is enzyme? You might wondering. So enzyme is proteins that control the rate of reaction in living things. So enzyme also are highly specific biological catalysts that act on specific reactant or substrate. Before we continue on, substrate is a substance acted upon by an enzyme, or simply say it is an ingredient for enzyme to make a product. So you might wondering, what is this thing that called catalyst? What does it mean though? So basically a catalyst is the thing that speed up reaction without being consumed in a process. So I have a graph here that could explain how the catalyst work. So this is a normal reaction without a catalyst or without an enzyme, and this is the reaction with catalyst or with enzyme. Notice the difference? The difference here that if a reaction doesn't get help or uncatalyzed by the enzyme, the normal reaction the activation energy will be higher, thus making the reaction to be longer to finish, while the one that getting catalyzed or getting helped by the enzyme, it'll be much more lower, thus making the reaction occurs more faster. Okay, so I have here some depiction of how enzyme work and how enzyme looks like. So this is a substrate, this is an enzyme active sites, and this is enzyme. Okay, the substrate or reactant move toward the enzyme's active sites. So it's moved to the active site. When it's already moved to the active site, the chemical reaction is triggered by the enzyme. When that reaction done, the enzyme releases the product. So the enzyme is a catalyst, which means it stays and it will be reusable when the next reaction occurs. So now there are some things that can affect enzymes' work. The first thing is the temperature. Every enzyme has an optimum temperature and a critical temperature. The optimum temperature is the temperature where the enzyme is mostly active, while the critical temperature is the temperature where the enzyme begins to denature. So denature itself means that it is a state when an enzyme has lost its original shape and structure. This has also become a reason why when you got a fever, your body need to get cooling down, so that the enzyme will not go denature and keep helping you with the reaction in your body to fight off the disease or something that causing the fever in your body. So here's the graph to make it more easier for you guys to understand. So if the environment of the enzyme is cold, it will not really active. It will not really active. But as the temperature rise into the optimum temperature, this is the peak of the enzyme performance. But if the temperature keep increasing beyond the optimum temperature, the enzyme will lose its activity or its performance and gradually and might become denature. The next is the pH effect on enzyme. pH affects enzyme much in the same way as temperature does. As the pH increases, the enzyme will reach its optimum point and function as fast as it can. However, as the pH continues to rise, the enzyme will be denatured and unable to work. And yeah, for example, if the enzyme in the intestine has optimum pH of 7.5, while those in the stomach have an optimum pH of 2. So here's the graph here. I'm going to show you. So from my explanation here, we can know that the activation, not the activation, the optimum pH of enzyme is depend on where it looks like and on where it's located. For example, as already said before, the enzyme in the intestine has an optimum pH of 7.5, while those in the stomach have an optimum pH of 2. As you can see here, if the pH is below the optimum pH, the enzyme will not really active, just like in the temperature. And as the pH increasing near to the optimum pH, it will also increase the enzyme activity. When it's reached the optimum pH, it is also reached the peak of its performance. But if the pH keep increasing, it will gradually lose its performance and activity and eventually become denatured. The next is substrate concentration. So the amounts of substrate in a reaction will obviously affect the amounts of product created. Once all the products are formed, no matter how long you wait, there won't be any more product unless you add more substrate. But on the other hand, if we continue to increase the amounts of substrate, eventually it will get to the point where all the enzymes are fully and working at their own max capacity. As you can see here, the reaction rate will keep rising as it should be, and if we keep increase the substrate concentration, it will keep increasing to the point where it will be stuck here, will not getting increasing anymore and just become plateau at this point, because the enzyme is already working at their maximum capacity. To make it more easier, we can let's imagine that we have a car factory. Boom, we have a car factory, and the worker in this car factory, let's say it's an enzyme. Boom, you have workers, and the spare part and the car and the car spare parts are the substrate. Boom, we have car spare parts and a box apparently. And for the product, we can imagine it as a car. There you go. So if you only have enough spare part to make one car, this tree worker will only make one car. Why? Because there is no other spare parts to making other cars. But let's say if we keep increasing the amounts of spare parts, boom, 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 boom. Okay, as you can see, if we keep increasing it, the worker might get overburdened or overloaded or overwhelmed, because they only can work at a pace to make one car and one spare part. With these lost spare parts, the other spare part will have to get queue before they can be turned into car, because the worker or the enzyme is busy with the substrate they're already on it. Enzyme name and the location. Okay, so I'll show you a graph, not a graph, a table here of where the digestive enzyme located. I know there's lots of enzyme in our body, there's lots of reaction in our body that needed enzyme, but if we consider it, there's like 1,300 maybe ish enzyme in our body. So yeah, we'll be focusing only on the digestive system enzyme. Okay, so the first is the mouth. In the mouth, it is the place where salivary amylase is located. What does salivary amylase do? It is break down starch into maltose. So this is the location. The next is stomach and pancreas, but we will get to stomach first. In the stomach, there's pepsin. What does pepsin do? It is break down proteins into dipeptides. Okay, next we'll go to the pancreas. In the pancreas, there are 3 enzymes located there. The first is amylase, second is trypsin, the third is lipase. Amylase, as it's already has the same name, and also it has the same function as the salivary amylase. It is to break down starch. Okay, the next is trypsin, which is also similar to pepsin. It is to break down protein into dipeptides. The difference here is trypsin are produced by pancreas into the small intestine to help the digestive process there. And the third is lipase. Its job is to break down lipids or fats. Okay, there's the location. And the next is small intestine. There are 4 enzymes that located in the small intestine. It is maltose, sucrase, lactase, and peptidase. Maltose jobs is to break maltose into glucose, while sucrase jobs is to break sucrose into glucose, while lactase jobs is to break lactose into glucose, while peptidase works is to break down dipeptides into amino acids. Okay, so that is the location of small intestine. Alright, I think that's all I can explain to you. Thank you very much for watching. If you want to know how is your understanding based on my video, you can check the link in the description. It will take you to the assessment page. There are some several questions there if you want to test how is your understanding. And once again, thank you so much for watching. Have a nice day, stay safe. My name is Mohamad Naufal Nursyabana. Wassalamualaikum warahmatullahi wabarakatuh.